Welcome to the second lecture in this series on how brains work from Frontiers of Science Online. In the first lecture, we talked about the human brain and how it worked, and we pointed out that brains are made up of nerve cells or neurons that are connected together as functional units, neural networks. And today we're going to be talking about neural networks for vocal communication. We also talked about two principles of brain function. One is what I call the real estate principle, location, location, and location. And that's the idea that different tasks that the brain is engaged in are handled in different brain regions, and that by learning about different regions of the brain, we can have some idea of how the very complex behaviors that the brain produces relate to the neural structures that are responsible. We also talked about the magnification principle, the idea that if a particular behavior or a particular function is very important to an animal, that part of the brain will be larger than it will in another species in which that function is not so important. So in us, for example, our visual areas are huge, as they are in all primates, and they're a lot smaller in a bat or a mouse. And today we're going to talk about areas that are involved in vocal communication, which are also very large in us and also overrepresented in species for which this is an important behavior. Now, the underlying assumption in all of these studies on how the brain works is the idea that you are your brain, what Francis Crick called the astonishing hypothesis, that you, all of your emotions, all of the very complex aspects that make you what you are actually represented in the nervous system. And I pointed out in the last lecture that this idea is actually a two-way street. So the brain both creates behaviors and is modified by the consequences of those behaviors. And one of the things that happens as the brain develops, because the brain doesn't know ahead of time what kind of organism it's going to end up in, is that experience modifies the brain and personalizes it. So your brain is different from the brain of everybody else on the planet as a result of your experience, both your learning experience and your cultural experience, but also the experience that happens inside of the body, the hormones that the brain is exposed to. So today we're going to talk about that idea, and we're going to talk about a significant aspect of personalization, how brains become male and female. We're going to talk about how you would discover exactly how that worked, how you take that system apart, and the idea of model organisms, systems in which a question of general interest can be studied rather easily. We're going to talk about a particular model organism, a frog that lives in Africa that sings underwater and sings its songs in the dark. And we're going to talk about how the male and female versions of the brain that produces those different songs is generated. And then finally, we're going to talk about the overarching issue of whether biology and destiny are inextricably intertwined, and how we might even look at that in an experimental way. So let's start out with the idea of personalizing your brain. So brains are very complicated. They're very complicated in both sexes. And the brain of a newborn doesn't actually know that much about the body in which it resides. And as a consequence of experiences that brain has both in utero and shortly after birth, and also at puberty, the brains of males and females become rather different. And one of the main ideas in this field is the idea that the experience that's responsible initially for making the brains of males and females different are hormones, hormones that are secreted by the male and female reproductive organs. So here's a diagram of one of those hormones shown up here. And these are steroid hormones, and we're going to talk about how steroid hormones sculpt the neural networks that are responsible for vocal behavior. So this is a talk about the vocal communication of sexual information, of information on reproductive status, on whether individuals are interested in mating. And we're going to talk about it in a particular model system. And this is the South African clawed frog, Xenopus laevis. Now let's talk for a moment about the frog, because all model organisms are also organisms with their own evolutionary history, their own trajectory, and their own intrinsic interest. So these guys live in sub-Saharan Africa, right below this cloud bank here. And they occupy this entire region. There are lots of different species of them. And as you'll see in a little while, they used to have a much larger distribution in Africa. But wherever you find standing bodies of water in Africa, you have a good chance of finding these frogs. So this frog, the South African clawed frog, you can see the little claws on the end of its toes, which it uses to reduce food to manageable proportions, has both males and females, as you would expect. And you can see here that the females are a lot larger than the males, a feature that I've always enjoyed in this species. And that's true in a lot of animals where a large part of the body mass of the female is made up of eggs. So the word Xenopus means strange foot, and the word Laevis means slippery. And these animals are so slippery that the males have developed a special mechanism that helps them hold on to the females. So here's a view of the underside of the frog. It's underwater, and you can see here gloving the inside of its arms and its fingers, black glove-like substance, which we call the nuptial pad. And if you go ahead and take a look close up using the scanning electron micrograph at what those nuptial pads are made up of, you see that they consist of these little epidermal spines. It's a little bit like Teflon. So in this magnification, this scale bar is 10 micrometers. There are 1,000 micrometers in a millimeter, so this is blown up quite substantially. You can't see these very well with your naked eye. Now, for our purposes, one of the good things about the animal is that it has distinctive male and female behaviors. So when a male is underwater at night, he sings, and he sings very frequently, sometimes for a half hour, an hour at a time, producing what we call the male advertisement call. Males produce this call by themselves, with other males, and if females are around, and it's advertising their presence to members of their own species, male or female. Now, females in this species come in two flavors. Most of the time, they are not ready to mate. And if a male clasps a female that's not ready to mate, one of the ways in which she communicates this is by using a specific behavior called ticking, which sounds like a clock. So ticking is a very effective behavior for males. They let her go. There's no percentage in hanging on to a female that doesn't want to mate with you. So one of the nice features of this system, just to begin with, is that the vocal behaviors of the sexes are very distinctive. The other nice feature is that we can represent the vocal behaviors very conveniently in a graphical form. So here's a graph, which you will recognize conceptually from Scientific Habits of Mind, our online text. And in the vertical axis is the frequency in kilohertz, 1,000 cycles per second. So this is the pitch. And on the horizontal axis is time. This is 1 second. And the male call, consists of trills that are made up of single clicks, where there's a fast part and a slow part, a fast part and then a slow part. Whereas female ticking is much slower. There are about 6 clicks a second, and she goes tick, 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 tick. So we have the great convenience in looking at the way the brain controls behavior, one of the things that we're interested in that we can represent the behavior itself very reliably in this graphical form. Now, these animals are extraordinarily successful evolutionarily. When we are long gone from the planet, when there are only cockroaches, these guys will probably still be around. And as you may remember, or will come to in the future from Peter Domenicol's lectures, we know that our Earth has seen only 5 major extinction events, one of which occurred here at the Tertiary Cretaceous boundary, and is believed to have been triggered by a meteorite hitting the Earth. There was an enormous loss of species diversity during the period that followed this meteorite, but the species, among the species that weren't lost, were the frogs that we work on. So you can trace the origin of these frogs back 200 million years, a point at which the continents were all put together, and the ancestor of these frogs migrated into Africa from Europe. Europe then separated out into a northern European portion and a southern African, South American, and Australian portion. And it was at this point, about 150 million years ago, that you first began to see direct ancestors of the animals that we're looking at. And then about 80 million years ago, we had the separation of the great landmass into the South American and the African continents. And there are ancestors of the species that went in both directions, some of which are still in Africa today, and some of which are studied by Linda Trueb at the University of Kansas, we can find in South America and up into the Caribbean. And then about 50 million years ago, you can see that what happens is the African continent starts to drift northward and dry out. But still at that point, these frogs were found in areas of the world that are very dry today, such as Yemen. And here are some fossilized animals from 50 million years ago whose skeletons look essentially identical to animals that we have today. And these have been studied by Amory Henrique and Ana Maria Baez. And so then today what we have is species of these frogs distributed throughout Africa with a close relative over there in South America. So why do we study these frogs? I mean, they're intrinsically interesting. It's fun to study an animal that's doing such a good job making it. But one of the reasons we want to study them is that they are a model organism. And a model organism is an organism whose experimental advantages are so great that progress in answering specific questions is accelerated. So what we want to know in these organisms is, among other things, what is it that occurs in the brain and in the body that produces such marked differences in the vocal behavior of males and females? And then at a higher level, we'd like to understand how the brain and behavior become coordinated in animals that are engaged in social communication. And you'll recognize here the fundamental assumption, which is that many of the mechanisms that we see in one organism, be it a fruit fly, a mouse, or a frog, are going to be very strongly conserved during evolution, because evolution is the great tinkerer. It very seldom throws everything away and starts over from scratch. It builds on what was there before. So if you're looking at an ancient organism that's working very well, the chances of its having answers to the questions that you're interested in some other organism are very high. So these frogs first came to the attention of modern day biology in the late 1930s because these were the animals in which the first pregnancy test was developed. And this test was popularized by this guy here, Sir Lancelot Hogben, an eccentric British scientist who was a great naturalist. And whenever he moved to a new position, and in this case he was moving from North America over to Africa, he'd go turn over the rocks, he'd find the native organisms, and he was very interested in hormones. And one of the things that he discovered was that these animals, if you injected them with urine from pregnant women, the females would lay eggs and the males would sing. And so that was the first pregnancy test, long before you had little strips of paper turning different colors. And that turned out to be very handy, because what it means is that at any time of year you can get the animals to sing. Normally in the wild they'll only sing for about half the year. In the lab you can get them to sing at any moment, and you can also produce embryos and get them to grow up. They're very handy, very hardy, very sturdy animal, incredibly sensitive to hormones, the same hormones in these frogs as we have in our own bodies. I put this slide up. It's actually a drawing that my daughter made when she was about 10 years old. And it brings up a point I think is very important, which is that even though you're using a model organism and you're studying questions of general interest, one of the things you always have to be sensitive to is the animal itself. If you have some kind of intuition about the animal, it often will help you in your experimental studies. So Danny said, after 20 years of biology, Dr. Kelley realized that she did not want to dissect a frog, but her destiny was to be one. And you see here she's put the little glasses on the frog. She's got the lab coat. Here's the wig for the hair of the frog. Identifying with your model organism can often be a great help in experimental studies. Okay, so we want to first of all see if they have sex-specific behaviors, which they do. They have the male advertisement call, which we heard before, and female ticking. So they have nice sex-specific behaviors that we can quantify using sound spectrograms. So how do they produce these songs? And they do it in an interesting way. Of course, they're singing underwater. And when we talk on expiration. Air has to flow out of our lungs in order to produce sounds. But these guys can't afford to breathe while they're singing, or they drown. So what they've done is taken their voice box, their larynx, and modified it so it can make sounds underwater. So here's the voice box. It's a cartilage box that is stiffened by rods and has muscle on the outside. And part of the sound producing apparatus is shown here in white. These are two discs. I'll show you the way it works in a moment, but one of the first things that we saw when we started working on the animal was that not only were the behaviors, the vocal behaviors of males and females different, but also the vocal organ itself was very different in the sexes. So the larynx is sexually dimorphic, is what we'd say, different in shape between males and females, and you can see that clearly here. So how are these little individual clicks produced? And it's actually pretty simple. The muscles contract, tendons pull on the discs, and every time the discs move, what happens is you get a single click sound. So here the discs have moved and produced a single click sound when they pop apart. So in a male, you can have click sounds as fast as 70 clicks a second, and in a female, it's an order of magnitude lower, only about 7 clicks a second. So this difference in the speed of contraction of the muscles is actually due to the kinds of muscle fibers that males and females have. Males have mostly fast muscle fibers, and females have mostly slow muscle fibers. So as a model organism, we can say, first, they have sex-specific behaviors, and second, the male and female vocal organs are also different in the sexes. So these are good places to start to try to figure out how the vocal system becomes sexually differentiated. So what about the brain? How does the brain work? And what we did was we actually took the model organism and made another model system within the model organism. Here's the brain of the frog. It's sitting in its head, and it connects up to its vocal organ. And the cells that go from the brain to the vocal organ are called motor neurons. They're nerve cells that sit in the brain and that send a process all the way down here to the muscle fibers. Here they're diagrammed, the motor neuron, the axon. And what these guys do is when they're electrically active, there's chemical information passed from the nerve cell to the muscle fiber. The muscle fiber then becomes electrically active and it contracts. And this connection between the motor nerve cell and the muscle fiber, which is shown down here, that connection is called the synapse, is called the neuromuscular synapse. And it actually turns out to be a very good model for the same kinds of connections that you see in the central nervous system. So in order to figure out where these cells are in the brain that are responsible for producing the sounds, what we did was to put small drop of dye on the axons, and this dye was then carried back by diffusion into the brain and showed us the neurons that are responsible for producing the clicks that produce songs. So here you see a side view of a frog brain. This is front and this is back, this is up and this is down. Here in this green area are located the yellow cells that are labeled with the dye, and these are the motor neurons that go and connect to the muscle fiber. So we can now ask the question of whether, in addition to the fact that there's a sex difference in the vocal organ itself, which you can see right here, here's the male larynx and the female larynx, we can actually also ask the question of whether the motor neurons are different in males and in females. So here are male motor neurons and here are female motor neurons, and I think even looking at this picture, you can tell that the male motor neurons are larger than most of the female motor neurons, and it looks like there are more of them. So it turns out there are about twice as many motor neurons in males as in females. So the model system has sex-specific behaviors, male and female vocal organs, and male and female brains. So what we want to know next, and the reason that we're using this model system, is how does this work? How does this come apart? And this is not an experiment that we get to do in other organisms that we dearly love. We wouldn't do it in chimps, we wouldn't do it in us. But on the principle that the same principles are going to apply, we can do it on these guys. So these are frogs, and like all frogs, they have two major stages of development. They're tadpoles for a while, and then they turn into small frogs, and the small frogs grow up to be bigger frogs. So these guys start out with fertilization. It takes them a couple months to get to the stage of metamorphosing from a tadpole into a small frog, and then about a year for a male to become sexually mature, and about 2 years for a female to become sexually mature. So what we can do is we can go and say, how do the sex differences in the motor neurons and in the muscle fibers develop? How do they come about? So what we discover is that if we look at males and females, they both start out with the same number of singing motor neurons, and then they start to die. They die in both males and females, but they die more rapidly, and there's more death in males than in females. So at the end, in females and in males, so that at the end of development, you end up with a situation where males have about twice the number of motor neurons as females do. So differences in the brain are due to less death in males, more death in females. How about the vocal organ? How does that work? Well, if you go back during tadpole life, they start out with the same number of muscle fibers. And then what happens is males start to add muscle fibers at a very rapid clip, so that by the time they reach sexual maturity, males have perhaps about 7 times more muscle fibers than females have, and it's quite a marked difference. So male numbers are greater than female numbers for the muscle fibers, not because fewer cells die, but because more cells are added. So there are 2 different kinds of ideas that are going on in males and females. There's the process that leads to differences in the brain where you have less death, and there's the process that leads to differences in the vocal organ where you have more cells formed. Okay, so we know now when all of these differences occur, and we know something about how, but what we don't know is what's the signal that tells a brain or a vocal organ to become male-like or female-like during development. So what we would say is, what's the mechanism? What's the biological mechanism? And we've known for a long time from studies actually in human beings that steroid hormones and the ability to respond to these steroid hormones are one of the key driving factors in how brains become male and female, in the sexual differentiation of the brain. And since this information was out there for mammals and for humans, we decided to go and see whether it was these hormones that were responsible for looking at differences in male and female vocal organs and in their motor neurons. So let me tell you a little bit about how these steroid hormones work, because they're not like a normal hormone. So let's take testes. The testes make a male sex hormone called testosterone. It's carried in the blood, bound to a protein, this brown substance here. And then the steroid comes off and it diffuses in the cell membrane. It actually dissolves in the cell membrane. It goes inside of the cell. It's picked up by a special protein called the androgen receptor. That whole complex is carried into the nucleus of the cell. 2 of these guys nestle down together on the DNA, bound to the hormone, the androgen receptor bound to the hormone, and they turn on gene transcription. And one of the reasons that we actually know this picture pretty well is that there are human mutants in which this system doesn't work. They have male sex hormone, but their receptors are not capable of turning on gene expression. So these individuals are often classified as female at birth. They grow up, they can't have kids, and it turns out that they're genetically male. They have testes, they make testosterone, but they can't respond to their own male sex hormone. So I think this is actually a very powerful example of the important role of steroid hormones. And a lot of the molecular biology of what we know about the receptor actually comes from the specific mutations in individuals with androgen insensitivity syndrome. So what we did in this study in order to determine whether the hormones were involved was we used drugs, anti-testosterones, that bound to the receptor but that could not turn on gene expression. And these drugs are, for example, used in prostate cancer, which is hormone sensitive. These are pretty powerful. They block the action of endogenous hormones. So what we did was we took the tadpoles and we grew them up either an anti-hormone or we didn't interfere with their development. And what you predict is that if the male sex hormone were involved, we should be able to reduce male motor neuron numbers to female levels. And you can see that that works in this slide. So here we're looking at tadpoles that were grown up either normally, so you're looking here at males and here at females, or in the presence of anti-testosterone. The name of the drug is hydroxyflutamide. So here's the normal sex difference in the number of motor neurons, and out here we can see that the number of motor neurons in males and in females is identical if we block the action of the sex hormone. How about the muscle fibers? The prediction would be if we gave females male sex hormone, they get more muscle fibers, or if we blocked hormone, we should be able to block muscle fiber addition. So here's an experiment where we gave female male sex hormone, we gave them testosterone, and you can see that the number of their muscle fibers, the error bars, again, scientific habits of mind, overlap in male and female. So we've been able to get rid of the normal male-female difference, and it turns out that you get rid of it because fewer cells die in females that are given male sex hormone. So in order for the hormone to work, you would expect that both the brain and the vocal organ would have receptors for the hormone, and you can actually go and take a look at that once you clone the gene for the receptor. And what you see here is a cross-section through the vocal organ, and here in this little itty-bitty piece of tissue right in here, you see a very bright area lighting up here and here. The bright area in the middle here are the stem cells that are going to make cartilage. So stem cells in the developing vocal organ are extremely sensitive to hormone because they've got lots of the receptor. And out here, this bright area is the developing muscle. And muscle stem cells, myoblasts, are extremely sensitive to developing sex hormone. And here in the brain, it turns out the expression of the receptor is like a stain for the whole vocal pathway. So here are the singing motor neurons. They were in green on the previous slide, and every single one of these singing motor neurons expresses the protein that enables them to respond to male sex hormone. So this is a good example of how two mechanisms, preventing cell death and stimulating the proliferation of muscle and cartilage stem cells, can lead to sex differences in the brain and in the vocal organ. Now, I'd like to actually just go in for a moment into the brain and talk about how the brain produces some of these vocal patterns and how you'd use a model organism to figure out how the brain makes songs that are different in males and females. And you'll recognize here that we're going after the astonishing hypothesis. We want to look at brain activity that's responsible for the production of a behavior. So one of the nice things about the frogs is that they're very strongly motivated to sing. So we can put electrodes around the nerve of the frog as it's singing. This is the nerve that goes to the vocal organ, and we can record the activity that's coming out from the brain while the animal is singing. And that's enabled us to do a lot of useful studies. But one of the things that's so extraordinary about it is that every burst of activity in the nerve matches a sound that's produced by the animal. So here we're looking at electrical activity in the nerve, down here in a female. So here's the nerve activity, and here's the sound that's produced on the upper trace, nerve activity and sound that's produced. So while this female is ticking, each tick is accompanied by a burst of nerve activity. And the pattern of the nerve activity perfectly matches the pattern that the sound is produced in. And in a more complicated way, we can do the same recording from the male while he's advertisement calling. And you can see that each burst of nerve activity is accompanied by a click. And as the bursts of nerve activity get louder, the clicks get louder, there's an exact match between what's coming out of the brain and the song that the animal is singing. So we use this method and other methods to go into the brain and to take a look at the neural networks that are responsible for the production of song. And we put dyes in various places. We saw how they were connected. We put recording electrodes in to look at electrical activity. And eventually we came up with this scheme here where you have the vocal motor neurons at the back of the brain, of the hindbrain. So those are the guys in green here. And then you have clusters of cells that are right here at the front of the hindbrain. So we identified two sets of circuits that were implicated in the control of the song: an anterior circuit, which includes this little area here, DTAM, in the anterior hindbrain, and a posterior or more further back circuit, the vocal motor neurons and also the vocal motor neuron target, which is the larynx itself. So we have two parts of the circuit, and what we'd like to know is whether each part of the circuit does something different. And at this point, we were able to take advantage of the very strong genetic control of behavior in Xenopus, and also of its incredible advantages as an experimental model organism in terms of how robust it is and how easy it is to deal with very early stages of development. So in Xenopus, each species has its own song. There are about 23 species that we know of. We discovered, we think, a new one a couple weeks ago. And each species has a very distinct song. So for example, this is one of the new ones that we just discovered that was just published, and we may have another one. So Laevis, you'll remember, goes like this. And then down here, here's another Xenopus species called Borealis that has a very different song. So these two songs are very different, and because they're very different, we can actually do an experiment that would be hard to do in other animals. We can switch the circuitry between two different species when the animals are in tadpole stages, and we can create what we call a chimera. So this is an animal that has part of its brain from one species and another part of its brain from another species. And let me just show you how you do that. So you take the embryo when it's about 24 hours old, before there are any nerve cells whatsoever, so there's no chance that it can feel any pain. And then what you do is you switch the front parts of the embryo, and you put the Borealis front hindbrain and the structures that are in that part on the Laevis back part, and vice versa. And you let these guys grow up, and you ask the question of what they sing. When they grow up, this is the way they look. They have part of them, the front part comes from Borealis. It turns out that the chimeras worked mostly in one direction. So here's two animals with front parts from Borealis. The skin is a little bit darker. This guy mostly has the skin around the eyes, and all the rest of the animal is Laevis. Actually, the vocal organ is also Borealis. So then we can ask these animals who want to sing when they're about a year old what kind of song they sing. Now, in order to do this experiment, you actually need a control, because it's possible that if they sing a goofy song, it was just because of the surgery that you used to create the chimera. So we also created chimeras between two different embryos that were both Laevis, or both Borealis, and the Laevis chimeras made songs that sounded like this. So they made a completely normal song. And the Borealis made songs that sound like this. So the transplants themselves didn't seem to affect the ability of the animals to sing when they grew up. So what about an animal that had the front part of the hindbrain circuit from Borealis and had the back part of the hindbrain circuit from Laevis. What did its song sound like? This is an experiment that was done in my lab by Brian Shaw. So in that animal, what you hear is a vocal pattern with an interclick interval. The one that predominates is identical to the borealis vocal pattern. There are these funky little extra clicks. We don't know where they come from. But the dominant pattern that's driving the production of the song in this animal is coming from borealis. So one interpretation of this experiment, it's not the only one, but it's a reasonable one, is that the pattern is created by the anterior circuits up here, but the rate is shaped by posterior circuits and the vocal muscles. And these animals actually had borealis muscles that can only contract rather slowly. So you have a complicated situation where you have the rhythm, which is generated by one portion of the circuit, and the ability to contract and relax rapidly, which is generated by the motor neurons and the hindbrain circuit. And this is a kind of taking apart of a neural circuit that's very hard to do in any other kind of system. So having taken the system apart, we thought it would be, at the end of apartheid, a good idea to go and see actually how it worked in Africa. How did the animals in Africa use these songs to communicate with each other? So my colleague Martha Tobias and I packed up our bags and went off to South Africa, to Cape Town in particular, where these animals come from. And we went there to see what kinds of songs males and females used when they were singing to each other in the dark. And what Martha would do, here's a picture of her out in a pond, is she'd go out and she'd look for ponds that had frogs in them by putting in these funnel traps baited with bones, and come back in the morning and see whether there are a lot of frogs. And then when we found those frogs, we'd go out and listen to them at night. And we made a number of very peculiar discoveries. So first thing we discovered was we could never see them, so we never knew who was calling. But unlike the situation here, you know, where you go out at night in the spring and there are 10 zillion frogs calling, in Africa we would only hear one animal calling at a time. It was very mysterious. But of course we didn't know who it was dark, they were down in the leaf litter. In order to figure out what was going on, what we ended up doing was bringing them back to the swimming pool in the house that Martha had rented for the summer and taking a look at what kinds of vocal communication they made in the swimming pool. And what we ended up doing out of desperation at the end of the summer was we put a male in this little portion of the swimming pool over here with a bone-baited funnel trap. And out here in this portion, we put a female. And when we did that, we discovered a new calling behavior that we'd never seen before. We discovered that when the female couldn't find the male, she started to produce a call called, which I'll show you in a second, called rapping, that was an incredible stimulant to the male. And I can show you this most easily and also point out the context in which these calls occur by showing you this movie. So here we're in the lab, and we've got a male clasping a female. And what's going to happen is he's going ar, ar, ar. She's going tick, 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 tick. What happens eventually is that her ticking suppresses his vocal behavior, so he shuts up. And then, just for a second, she calls a little bit faster, and he gets all excited again. She goes back to ticking, and he shuts up again. Okay, so now he's shut up. Now a little bit faster. You heard that little tiny burst? He got all excited again. She's back to ticking. He shuts up again. So what we discovered by going to Africa was not only a new call that females made, not just ticking, which you know before, but also a new social interaction. So here's ticking, which is the female call that we had studied before. And here's the call that we first heard in a female where she couldn't find a male when she was about to lay her eggs. And we call this call rapping. Sounds like machine gun fire, rapid Geiger counter. So these calls have very different effects on the male's behavior. What happens to a male that hears ticking is that he shuts up. So that's vocal suppression. And rapping, on the other hand, functions as a vocal aphrodisiac. And if you play rapping to a male underwater from an underwater loudspeaker, one of these pool loudspeakers, the male will swim over to the loudspeaker and try to mate with the loudspeaker. Nothing about the evidence of his other senses, the fact that he's touching metal, the fact that there's no female there, nothing about that deters him from the excitement that's produced by female calling. So I mentioned a minute ago that when we went out to the ponds, we only often heard one male calling, and yet when we pulled those funnel traps out of the ponds, there were thousands and thousands of males in a particular pond. So how come only one male calls at a time? So when we brought the animals back to the lab, we began to have some insight into that. And let me just show you how that works. And you'll notice how much more energetic this interaction between the males is than the interaction between the males and the females. So here's a female and here are two males, and we're going to see them calling. We're going to see them ignore the female, and we're going to see them interacting in a very vigorous vocal fashion. That's chirping that one of the males is doing. Okay, now they face off. One of the males starts ad calling. This guy comes over and starts clasping him. You get chirping and growling, chirping and growling. So males also have vocal suppression. And you can take two males that were calling for equivalent lengths of time before you put them together. When you put them together, both of them call less. You can see the vertical axis here. This one starts at 3,000, this one starts at 1,000. But one of them shuts up entirely. It becomes vocally subordinate. And then if you remove the dominant male, the subordinate male goes back to calling. So both males and females have these highly specific vocal interactions. And all of their vocal communication is occurring within a very specific context. So if you look at the total number of calls that they produce, males produce 6 calls. The advertisement call, the answer call, that really excited one when they hear the other animal, chirping that you heard, chirp, 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 growling, when they're classed by an animal, and plectin calling, da-da, 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 when they're clasping another animal. And females produce these 2 calls, ticking, get off my back, and rapping, the context of which is, I'm about to lay my eggs, where are you? If you look when males call to each other, they use all 6 of these calls. They have a very vigorous, very rich social interaction. They're using their songs in a particular social context. When they call to females, on the other hand, they only use 3 of them. So they're actually having a more complex vocal interaction with females. And surprisingly to me, when you put 2 females together, you don't get any vocal behavior out of them whatsoever, although they will occasionally fight if they're both receptive and there's a male in the tank with them. So these animals have a fantastically useful system for dealing with life underwater in the dark. Males probably are struggling with each other, facing off on a female. One of them will win and become vocally dominant, probably displacing the other male. And the way we think that it happens in the ponds is that they live together in these crowded, murky ponds in South Africa. And only one female becomes sexually receptive at any time. So the males are always ready to go, but there are very few females that are ready to lay their eggs. So here's a female that's sexually receptive. Here are all these males ready to go, but only one target for them. The singing male silences all of the other males. The female's attracted to the calling male. She raps, he calls, they get together, and the payoff is the production of tadpoles and the population of the ponds. So this, I think, is actually an example of the magnification principle that we talked about in the first lecture. Here's an animal whose entire life is acoustic, whose entire form of social communication, aside from clasping each other, lies in the vocal realm. And in this animal, the way that the nervous system is connected to connect what an animal hears and what an animal produces is very highly exaggerated and very powerful. And it makes this kind of a system very good, not only for discovering what it is that drives differences in the ability of males and females to make different vocal patterns, what it is genetically that drives the different vocal patterns across the species, but also what drives the ability of two brains to communicate with each other, which is going to be the topic of the third lecture in this series on the evolution of language. Thank you.